Students, already in your lower class in the 10th, in the chapter Genetics and Evolution, right, you studied about the different experiments performed by Gregor John Mendel, like monohybrid cross, as well as one more is dihybrid cross. In second PU, in this chapter, once again you are going to discuss the same but little in detail. Here, apart from monohybrid cross, dihybrid cross, you are going to discuss few other different crosses also. Okay, so whichever the experiment of Mendel it may be a monohybrid, it may be a dihybrid, whichever it may be, there are few basic steps which are common in all experiment. Okay, in my next lecture, I am going to discuss about monohybrid and dihybrid and few more experiments. ಯಾವ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಬೇಕಿದ್ರೂ ಆಗಿರಲಿ, ನೀವು ಅದನ್ನು ಸ್ಟಡಿ ಮಾಡುವಾಗ, ಓಕೆ, ವೆನ್ ಯು ಗೋ ಥ್ರೂ ದ್ಯಾಟ್, few basic steps are very common. So let us see what are those. If you understand those basic techniques, or I have written here Mendel's experimental technique, those basic techniques, if you understand, the same thing you are going to write again in the upcoming monohybrid, dihybrid and other crosses. Is that clear? Okay, stay focused. Now, what are the experimental techniques of Mendel? It include few steps or the few techniques. Among them, the first one is selection of, what is the first one? Selection of characters. Write down. Have you written the heading? Next, okay, I will dictate. Meanwhile, you have to write down what points I dictate. Write down the same what I told. Okay, now number one, step number one, selection of characters, or you write down contrasting characters in Pisum sativum. Okay, Pisum sativum. Now, what do you mean by characters? I again told you, it is not good or bad character. Here I am going to discuss about different, which one? Genetic character. What do you mean by a character? Character is a well-defined morphological feature of an organism. Listen to me carefully. What is character? Character is a well-defined morphological feature of an organism. ಒಂದು organism in a clearly visible feature. When I look at you guys, when you are in my class, when I look at you, a very clearly visible, imagine yourself in the class, all of you friends are together there. When I look at you, the clearly visible morphological feature, ನಿಮ್ಮನ್ನ ನೋಡಿದ ತಕ್ಷಣ ಹೇಳುವ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಏನು ಅಂತ ಕೇಳಿದ್ರೆ height. Okay, ನಿಮ್ಮ height. I am not mentioning here tall or dwarf. I am just saying height. I say hairstyle. I am not saying straight hair, curly hair. Okay, I am just mentioning here what is a character? Hairstyle. Eye color. I am not saying blue eye, brown eye, black eye. No. So well defined morphological feature of an organism. ಕಣ್ಣಿಗೆ ಕ್ಲಿಯರ್ ಆಗಿ ಕಾಣಿಸುವ ಒಂದು ಎಕ್ಸ್ಟರ್ನಲ್ ಫೀಚರ್. ಓಕೆ, ಎಕ್ಸಾಂಪಲ್ ಯಾವುದಾಗಿರ್ಬೋದು? ಹೈಟ್, ನಾಟ್ ಟಾಲ್ ಅಂಡ್ ಡಾರ್ಫ್, ಅದು ಮತ್ತೆ ಹೇಳ್ತೇನೆ. ಹೈಟ್, ಹೇರ್ ಸ್ಟೈಲ್, ಐ ಕಲರ್, skin color, right? All of them are the clearly visible features which are called genetic characters or the clearly visible characters. So, how many characters he selected? He mainly selected 7 characters. So, how many characters he selected? Totally 7 characters he selected. What are they? They include, the first one is stem height. What is the first character? He selected stem height as a character. When he observed in the various Pisum sativum plants and he considered stem height as a character, he come to know that some of the pea plants were, okay, some of the pea plants were too tall, some were what? Dwarf. And he come to know that Out of the two, one is dominant based on his study. I will tell you the meaning, okay? Another one is what? Recessive. Clear? Okay. So here, first he selected character. How many character he selected? 7 character. For each character, he selected its what? Traits. He selected its traits. What do you mean by trait? Right? What are traits? It is the distinguishing feature of a character. Try to understand. I already told you the meaning of character. Well defined or clearly visible morphological feature of an organism is called character. Alva? Right? Example, height anta helidri. Distinguishing feature of a character. ಅಂದರೆ ಒಂದು ಕ್ಯಾರೆಕ್ಟರನ್ನೇ ನೀವು ಡಿಫರೆನ್ಶಿಯೇಟ್ ಮಾಡ್ತಾ ಇದ್ದೀರಿ. ಡಿಸ್ಟಿಂಗ್ವಿಷಿಂಗ್ ಫೀಚರ್ ಆಫ್ ಅ ಕ್ಯಾರೆಕ್ಟರ್ ಕ್ಯಾನ್ ಬಿ ಕಾಲ್ಡ್ ಟ್ರೇಟ್. ಸೊ ಕ್ಯಾರೆಕ್ಟರ್ ಮತ್ತು ಟ್ರೇಟ್ ಎರಡು ಮೀನಿಂಗ್ ಸೇಮ್ ಅಲ್ಲ. ಒಂದು ಕ್ಯಾರೆಕ್ಟರನ್ನು ನೀವು ಡಿಸ್ಟಿಂಗ್ವಿಷ್ ಮಾಡಿದರೆ, ಎಕ್ಸಾಂಪಲ್ ಫಾರ್ ಅ ಕ್ಯಾರೆಕ್ಟರ್ ಈಸ್ height. Height can be distinguished into tall and dwarf. Now, very simple. Hairstyle is a character or a trait. It is a character. I can distinguish the hairstyle among your class members into straight hair, curly hair, some other, right? Distinguishing feature. Eye color is a quick, very simple character. Blue eye, right? What is that? Black eye or the brown eye are its traits. ಇನ್ನೊಮ್ಮೆ ಹೇಳ್ಬೋದಾ? Recall ಮಾಡ್ತೀರಾ? Very simple. What are character? Practice ಮಾಡಿ. Well defined morphological feature of an organism. Example, height, that I will dictate later. Okay. What are traits? Distinguishing feature of a character can be called traits. Example, tall and dwarf. Tall and dwarf trait. Height is a character. ಈ ಎಕ್ಸಾಂಪಲ್ ಅನ್ನು ನೀವು ಕರೆಕ್ಟ್ ಆಗಿ ಜ್ಞಾಪಕ ಇಟ್ಕೊಂಡ್ರೆ, ಇಫ್ ಯು ರಿಮೆಂಬರ್ ದಿಸ್, ದ ಡೆಫಿನೇಷನ್ ಯು ಕ್ಯಾನ್ ರೀಕಾಲ್ ಬೈ ಯುವರ್ ಓನ್. ಯು ಕ್ಯಾನ್ ರೈಟ್ ಯುವರ್ ಓನ್ ಡೆಫಿನೇಷನ್. ಇಟ್ ಈಸ್ ವೆರಿ ಸಿಂಪಲ್. ಓಕೆ, ಫಸ್ಟ್ ಕ್ಯಾರೆಕ್ಟರ್ ದಟ್ he selected was which one? Stem height. Stem height was available in how many traits? 2 traits. One was tall, another one was dwarf, and it is represented by capital T. Tall is a dominant one. Dwarf is a recessive, meaning we will see later. You might be knowing that. Okay. Now, tall is controlled by a gene or a factor called capital T. Dwarf is controlled by small t. Be careful. When you use the uppercase or the capital letter for the dominant, right, dominant trait for a character, for its recessive trait, its small letter must be used. Here capital T, here you can't use capital D or small d. ಒಂದು ಕ್ಯಾರೆಕ್ಟರಿನ ಎರಡು ಟ್ರೇಟ್ಸಿಗೆ, right, for dominant trait capital letter, for recessive trait small letter you are going to use. Okay, next, which one? Flower position. You have to remember all the 7. Very simple. Flower position, they may be either axillary in position, represented by A, or control the gene A, or in some ಅವರು ಏನಾಗಿದ್ರು terminal. What is that? Small a. Flower position may be either axillary or they may be terminal. The third one is number 3, flower color. Okay, they were available mainly in 2 colors. One is which one? Violet, capital V. The recessive was white. Shall I write W? No, small v. Okay, flower position, flower color was the 2 more characters what he selected. The 4th one is seed, shape. 5th one is seed colors. Okay, seeds were available in 2 shapes. What are they? Some seeds were round, some seeds were what? Wrinkled. So seed shape, some are round, okay, some are wrinkled. Be focused. Okay, you should be able to differentiate which one is a dominant trait, which one is a recessive. Seed color, it was yellow was the dominant, okay, green was what? Recessive. Seed color yellow, green. Apart from that, the two more characters that he selected was, one more was pod shape, and the seventh one is pod color. Okay, pod shape. If you observe the pod, okay, some were how inflated, some are, I am not drawing the chili here, okay, ಮೆಣಸಿನಕಾಯಿ draw ಮಾಡ್ತಾ ಇಲ್ಲ. I hope you are following, go through NCERT diagram otherwise, okay. In some, the pods were inflated. ಆ ಪಾಡಿನ ಒಳಗಡೆ ಏನಿದೆ ರೀ, ಸೀಡ್ಸ್. ಆ ಸೀಡ್ಸ್ ಹೇಗಿರ್ಬೋದು, ಐದರ್ ರೌಂಡ್ ಇರ್ಬೋದು ಅಥವಾ wrinkled ಆಗಿರ್ಬೋದು ಅಲ್ವಾ. ಪಾಡಿನ ಒಳಗಡೆ ಏನಿದೆ, seed will be present. Hope you know that. If you observe the shape of the pod, some case they are what? Inflated. Or in some case they are not inflated, they are constricted, which is the letter small I. Inflated pod, constricted pod. Another one was pod color. The pod color was green, which was dominant, yellow, which was recessive. The major mistake that you usually do is, okay, you will get confused between pod color and seed color. Always remember, seed color is yellow green, yellow dominant, green recessive. Pod color green yellow, green is dominant in the pod color, yellow coloration is what? Recessive. Don't commit mistake, don't get confused between seed color, pod color. Another most common mistake that you used to do is flower color. What you write? White violet you write. No, violet is dominant, white is recessive. So by this, what is the first basic technique employed by the Mendel to that Pisum sativum plant? From that plant, by his clear observation, he selected how many character? 7 character, along with each character's what? Traits, along with its contrasting traits, with its contrasting traits. Is that clear? So 7 character, totally how many trait? 14 traits. This is the first step. Write down, complete this one under the step number 1. Once you do this, now next, number 2. Okay, number 2 is, you can write down, obtaining the true breeding plants. Obtaining the true breeding plants. Write down. Explanation I will dictate. Listen to me carefully. Once he selected character and its trait, second step ಅವನು ಗಾರ್ಡನ್ ಅಲ್ಲಿ ಮಾಡಿದ್ದು ಏನು ಅಂತ ಕೇಳಿದ್ರೆ, ಫಾರ್ ಈಚ್ ಟ್ರೇಟ್, ಪ್ರತಿ ಟ್ರೇಟಿಗೂ ಕೂಡ he obtained true breeding plant. What do you mean by true breeding plant? Are those which are genetically pure. True breeding plant andre enu? The plants which are genetically pure are called true breeding. Nothing but. It can be defined as the plant upon undergoing continuous self-pollination will transmit the stable trait inheritance is called a true breeding plant. Very simple, you will understand the definition. Listen to me. For example, he took which plant? Tall plant ಅನ್ನು ತಗೊಂಡ ಅಂತ ಅಂದ್ಕೊಳ್ಳುವ. ಈ ಟಾಲ್ ಪ್ಲಾಂಟ್ ಅನ್ನು ಅವನು ಏನು ಮಾಡ್ತಾನೆ? What he will do? Self-pollination. Upon self-pollination, what he got its next generation, if it is also what? Tall. Now, when these are self-pollinated, again their next generation is what? Tall. So, What do you mean by true breeding plant? A plant upon undergoing continuous what? Cross-pollination or self-pollination? Continuous self-pollination will show stable trait inheritance. ಮತ್ತೆ self-pollination ಆದವಾಗ will show stable trait inheritance. The same trait, the tall nature will be inherited. Stable trait inheritance to the successive generation is called a true breeding plant. 1 mark, very important question. Can you recall the definition once again? A plant upon undergoing continuous self-pollination. Dear student, here you cannot by heart and write in the exam. Genetics, mol bio, evolution, by mug up you cannot achieve the success. Lot of concepts are involved here. You need lot of patience as well as practice. Without practice you can't understand this concept. So stay focused, practice along with me, understand the meaning, understand the concept. Clear? Now, a plant upon undergoing what? Continuous self-pollination will show stable trait inheritance to their successive generation are called true breeding plants. In his garden, he obtained true breeding plant for each trait. At one side of the corner of his garden, there was a few plants which were tall true breeding plant. At another side, dwarf true breeding plant. One more side, axillary flower position true breeding plant. Hope you are understanding. For each trait, he obtained what? True breeding plant. Now my question is, in his garden, how many variety of true breeding plants are present? How many variety? 14 variety of, right? 7 character, 14 traits. Each trait true breeding plant ಅಲ್ವಾ? ಪ್ರತಿ tall true breeding plant ಇದೆ, dwarf true breeding plant ಇದೆ. ಪ್ರತಿಯೊಂದಕ್ಕೂ ಕೂಡ ಈ ರೀತಿ ಮತ್ತೆ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಮಾಡಿ ಅದರ ಜೆನೆಟಿಕಲಿ ಪ್ಯೂರ್ ಫಾರ್ಮ್ ಅನ್ನು ಅವನು ಆಪ್ಟೇನ್ ಮಾಡ್ತಾನೆ. 1 mark question, in the garden of Mendel, how many true breeding variety plants were there? Your answer is very simple. For each trait true breeding means, what is your answer? 14 varieties of true breeding plants were present. Write down. Number 2, obtaining the true breeding plants. In that, write down. True breeding plants are those plants. True breeding plants are those plants. Underline true breeding plant. Mark it as a very important 1 mark question. Are those plants which upon undergoing continuous self-pollination will show stable trait inheritance to the successive generation. Okay, this is a 1 mark question. Define true breeding plant. Write the standard NCERT definition. Next line. They are also called, they are genetically pure. Next line. True breeding plants are what? Genetically pure. Underline genetically pure. Next line. Totally 14 variety of, underline 14 variety. Totally 14 variety of true breeding plants were present in the garden of Mendel. Underline 14 varieties. So dear student, first he selected character and its trait. Practice along with me. For each trait he obtained what? True breeding plant, which was his second step. What is his third step? It is hybridization of, write down, hybridization of the true breeding plants. Third step is hybridization of the true breeding plants. What does this means? What he did is he selected a character, say for example height as a character. Already in his garden the true breeding tall plant is present, true breeding dwarf plant is present. He selected a character and its true breeding traits which are there, right? The traits which are under genetically pure or the true breeding plants, they were crossed. Say for example, just understanding in detail we will see later. This is a pea plant which is tall, okay. This is a dwarf pea plant. Tall true breeding plant, dwarf true breeding plant he selected. Suppose if he consider flower position, he consider axillary true breeding plant, terminal flower position true breeding plant. He cannot cross a axillary true breeding plant with a dwarf true breeding plant. ಅವನು ಎಕ್ಸ್ಪೆಕ್ಟ್ ಮಾಡುವ ರಿಸಲ್ಟ್ ಸಿಗೋದಿಲ್ಲ. ಆ ಕ್ಯಾರೆಕ್ಟರಿನ ಎರಡು ಟ್ರೇಟ್ಸ್ ಇದೆಯಲ್ವಾ, ಅದನ್ನೇ ನೀವು ಏನು ಮಾಡಬೇಕು, ಕ್ರಾಸ್ ಮಾಡಬೇಕು. he cross pollinated or self pollinated? He cross pollinated. Remember what he did? Cross pollination. Okay, in the first step he selected the two parents, then he did cross pollination. How he performed cross pollination? What he did during cross pollination, he conducted emasculation. Okay, emasculation, dusting as well as bagging. Very simple. During cross pollination he did emasculation. What is emasculation? As you know, it is a bisexual plant, right? A single plant itself, its flower has both male gamete in the form of a pollen, right? In the anther pollens are there, even a female gamete, ovule or the ovary what we say, that is also present here. It is a bisexual. If you allow that plant as such, what will happen? Its flower, what is there, no? Imagine it is its flower. Okay, here is the stigma, sorry, the style. Here you have stigma. Okay, this is the stamen, here is the anther. Now it is a bisexual plant. If you allow it as such, its own pollen may germinate its own female gamete. Then it becomes what? Self pollination. ಆದರೆ ನಿಮ್ಮ ಟಾರ್ಗೆಟ್ ಏನು ಇಲ್ಲಿ? ನಿಮ್ಮ ಟಾರ್ಗೆಟ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್. ಟು ಅಚೀವ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್, what you are doing? Emasculation. What is emasculation? It is the removal of stamens. It is the removal of stamens. Okay, then its pollens which are there, those pollens are dusted on, okay, the stigma of the another plant. Its pollen will be collected with the help of a brush. The stamen will be cut, the pollens are collected with the help of a brush. Those pollens are dusted on the stigma of the another plant. Its stamen, what you have cut already, the pollen from the another plant, its pollens are dusted on the stigma of the another parent. So that is what we call emasculation, is the removal of what? Anther, to avoid self pollination and to achieve cross pollination. Later pollens are dusted on the stigma. After that, what you will do? You will put a bag on it, plastic bag, a cover on it, a paper cover will be placed on it. That is called bagging. Why you are doing the bagging? To avoid pollination by, right, pollination contamination. Okay, what you will do? Bagging. So hybridization of the true breeding plant, where you are selecting the two true breeding plant, you are doing cross pollination with the help of emasculation, dusting and bagging, due to which you will get what? Its progeny or the first generation. And the first generation, what you get, that generation is called F1. What do you mean by F1? Filial one generation. Okay, here I'm going to use the term F1. F1 refers to filial one generation. I don't know, a plant, a baby plant or the first generation plant is obtained. This F1 plant, usually what he will do is, he will do what? Self-pollination. Once he get F1, the F1 plant will be self-pollinated, because of which he is going to get its next generation, that is called F2. What do you mean by F2? It is the filial 2 generation. I don't know, here he is going to get what? Tall, dwarf variety of plants. So I repeat, usually he select the true breeding plant. First he will do cross pollination. He will get F1 generation. F1 plants are self pollinated to get F2 generation. You may ask me, why here it is cross, why are it is self pollination? Definitely you are going to understand this when I start the concept monohybrid and the dihybrid cross. Okay, ಆವಾಗ ನಿಮಗೆ ಸ್ಪಷ್ಟವಾಗಿ ಹೇಳ್ತೇನೆ ಯಾಕೆ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್, ಯಾಕೆ ಇಲ್ಲಿ ಸೆಕೆಂಡ್ ಟೈಮ್ ಎಸ್ಪೆಷಲಿ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್. ಫಸ್ಟ್ ಟೈಮ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಇಸ್ ಮ್ಯಾಂಡೇಟರಿ ಬಿಕಾಸ್ ಯು ಆರ್ ಡೂಯಿಂಗ್ ಹೈಬ್ರಿಡೈಸೇಷನ್. ನೀವು ಒಂದು ವೇಳೆ ಫಸ್ಟ್ ಟೈಮ್ ಕೂಡ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ನೇ ಮಾಡ್ತಾ ಕೂತ್ಕೊಂಡ್ರೆ ಅಲ್ವಾ, ಅದೇನಾಗುತ್ತೆ, ಇಟ್ ಈಸ್ ಆಬ್ಟೈನಿಂಗ್ ಅ ಟ್ರೂ ಬ್ರೀಡಿಂಗ್ ಪ್ಲಾಂಟ್. ಅದರ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ವೆನ್ ಅ ಕಪಲ್ಸ್ ರೈಟ್ ದೇ ಗಿವ್ ಬರ್ತ್ ಟು ಅ ಪ್ರೊಜೆನಿ, ವೈ ದೇ ಆರ್ ಡಿಫರೆಂಟ್, ಅದನ್ನು ನಾವು ಚೆಕ್ ಮಾಡಲಿಕ್ಕೆ ಟ್ರೈ ಮಾಡ್ತಿದ್ದೇವೆ. ಹಾಗಾಗಿ ವಾಟ್ ಈಸ್ ಮ್ಯಾಂಡೇಟರಿ? Hybridization is mandatory. This is how he did what? Obtained F1 generation. From F1 by self-pollination, he obtained F2 generation. I will dictate the point. Listen to me. And the fourth step in the experiment was he maintained a neat, a proper record of the results what he obtained in his each experiment. Every time he did experiment with a tall dwarf, axillary terminal, round wrinkled, yellow green, with everything, whatever result he got, he maintained the neat record of those results. Those results 1900 onward that helped the three rediscoverers. Who are they? Hugo de Roys, Carl Correns, ಎರಿಕ್ ವಾನ್ ಶಮಾಕ್. ಅವರಿಗೆ ಹೆಲ್ಪ್ ಆಗಿದ್ದು ಅವನು ನೀಟಾಗಿ ಮೇಂಟೈನ್ ಮಾಡಿದ ರೆಕಾರ್ಡ್ ಅಲ್ವಾ. ನೀವು ಕೂಡ ಹೇಗೆ ಫಸ್ಟ್ ಇಯರಲ್ಲಿ ರೆಕಾರ್ಡ್ ಬುಕ್ಕನ್ನು ತುಂಬ ನೀಟಾಗಿ ಮೇಂಟೈನ್ ಮಾಡಿದ್ರಿ, ಐ ಹೋಪ್ ಸೆಕೆಂಡ್ ಇಯರಲ್ಲಿ ಈ ವಿಡಿಯೋ ಲೆಕ್ಚರ್ಸ್ ಏನು ಆಗ್ತಾ ಇದೆ, ಐ ಹೋಪ್ ದ್ಯಾಟ್ and I'm right, I hope that you are maintaining a neat notes of it like your friend Mendel. Is that clear? This is how the 4 basic steps are employed by Mendel in his almost all experiment. The same thing you are going to use it. Write down. Step number 3. In step number 3, write down hybridization of true breeding plant. Next line in that, write down. Next line. Mendel crossed, underline crossed, true breeding plants to get filial 1 generation, underline in bracket F1, filial 1 generation, full stop. Continue the line. F1 plants were self-pollinated, underline self-pollinated. F1 plants were self-pollinated to get F2 generation, Next line. During crossing, or you write down, during cross-pollination, he conducted emasculation, underline emasculation, in bracket removal of stamen of a bisexual flower to prevent self-pollination. Close the bracket, comma, dusting, underline, and bagging. During cross-pollination, he performed emasculation, dusting, and bagging. Underline all the three. Step number 4. He maintained neat record of results of his experiment. Is it clear? So these are the 4 basic experimental techniques which I am going to apply in monohybrid as well as dihybrid cross. Okay, in my next lecture I will dictate the definition of character, trait, as well as we are going to discuss other genetic terminologies.